This is your RNA and protein synthesis set of notes. So let's start off here by remembering that DNA is the molecule, the macromolecule. It's a nucleic acid in the nucleus that contains all the instructions to make proteins. And the sequence of nucleotides is specifically what codes for that protein. And depending on the sequence of nucleotides, you can get different proteins. And obviously, the sequence of nucleotides is the reason why individuals are different. A gene, and you do need to know this vocab, a gene is a sequence of nucleotides in the DNA molecule that can be hundreds of letters long, it could be thousands of letters long, but the point is it codes for a protein. And remember that ultimately we have to get from DNA two proteins, which means we have to somehow go from DNA to ordering amino acids and putting them all together to make proteins. So that's what this set of notes is about. And that's why we talk about RNA. So very briefly here, RNA is ribonucleic acid. The difference between that and DNA is the ribose, right? Ribose is the sugar in RNA, whereas deoxyribose was the sugar in DNA, right? And again, it is a macromolecule, it is a nucleic acid, right? Just like in DNA, the building blocks for RNA, the monomers, are nucleotides, nothing new, right? Some differences are that RNA is single-stranded. There's only one strand in an RNA molecule, and it can leave the nucleus. It is not trapped in the nucleus like DNA is. So let's look at the nucleotides for RNA. It looks the exact same. You have a phosphate, you have a 5-carbon pentose sugar, you have a nitrogenous base, but again, the difference here is that the sugar is ribose, right? Ribose means sugar, whereas in DNA it was deoxyribose. And the nitrogenous bases are almost identical with the one exception of uracil, that should be an I. Uracil replaces thymine. So notice you do not have T's in RNA molecules. That's a giveaway. If you ever see a U, you know you're looking at an RNA molecule. There are 3 types of RNA, which we'll talk about in turn. The first is messenger, that's what the M stands for. The second is transfer, that's what the T stands for. And the third is ribosomal, which is actually what ribosomes are made of, which is what the R stands for. All right, messenger RNA, it tells you exactly what it does. It is the messenger. It carries information from DNA in the nucleus to ribosomes in the cytoplasm. This is how the information exits the nucleus, right, via messenger RNA. And on the messenger RNA, we talk about these nucleotides in sets of 3. So let's look at this messenger RNA strand over here. Notice that it is single-stranded, which tells you it's an RNA molecule. There are U's in here, right? Which is the second indicator that this is an RNA molecule. There are no thymines, there are no T's. And other than that, you wouldn't know that this is RNA, but those are the 2 giveaways. And if you read them in groups of 3, almost like you would read words in a paragraph, right? If you think of these letters as letters and these sets of 3 as words, these are called codons. And you do need to know what a codon is. It's a sequence of 3 nucleotides in a messenger RNA strand. In a messenger RNA strand. Each codon codes for an amino acid. So codon number 1 is specific for a certain amino acid. I'm going to abbreviate those AA. Codon number 2 is also specific to a different amino acid, possibly, right? So each codon is going to request a different amino acid. So you can start to see how we're leading up to making proteins here. The next player in this game is transfer RNA. The job of transfer RNA is to, you guessed it, transport or transfer the amino acids that are in the cytoplasm to the growing protein at the ribosome. So the transfer RNA kind of has this little loopy, these are called hairpin loops, section, and then the amino acids up here on the top. And down here on the bottom is I would say, the most important part. It's called the anticodon. The anticodon. And the whole point of the anticodon is to match the codon that's on the messenger RNA. So let's say here's a messenger RNA strand that has U, A, C. That is the codon. What would the matching anticodon be? Well, the matching anticodon would be A, U, G, right? Remember that there's no thymine, so you would want to put a T here, but remember, this is an RNA molecule, so it's a U. All right, the last player here is rRNA. Remember, R stands for ribosomal, and that's because ribosomes are actually made of RNA. They're made of rRNA. So here's a computer-generated model of a ribosome waving at you. It's just kind of a cute little picture. They are 3D structures. I just want you to realize that in our images, they're going to be flat and 2D, but in real life, obviously, they have a 3D, a third dimension to them. What's going to happen is that messenger RNA is coming from the nucleus. Its destination is the ribosome. And at the ribosome, it's going to meet with tRNA, and the ribosome is going to start to putting the amino acids together. The ribosome is basically like an enzyme. It has a similar function. It's catalyzing the reaction between the amino acids to link them together. So before we talk about how all that's done, let's do a quick recap. So DNA versus RNA. You can pause this. Make sure you understand this. This is basic knowledge, right? Double-stranded versus single-stranded. What the sugar is. That the bases are slightly different. T and a U. DNA is in the nucleus. RNA can leave. And there are 3 types of RNA here, right? All right, so let's tackle protein synthesis. How do you make proteins? So there are 2 main steps down here at the bottom. We're going to do each of these in turn. The first is transcription, and the second is translation. And you can remember the order because alphabetically C comes before L. That's kind of one way to do it. All right, so let's look at transcription first. You're going from DNA to messenger RNA. That's critical. You must know it, right? So transcription is the process of making a messenger RNA from a DNA template. So if you look in this image, what I'm highlighting here in red is the original DNA in the nucleus. And you can tell, first of all, because it's double-stranded. Second of all, because you've got T's in there. You know it has to be DNA. This third strand that I'm going to label number 3, right here, is the RNA strand. And you can tell it's RNA because you've got uracil. You've got U's rather than thymines, right? So it's very, very similar to DNA replication that we talked about in the last video. The only difference is that here you're making a messenger RNA strand using uracil, and obviously the sugars in this strand are ribose sugars. So just like DNA duplication or replication, you have to unzip the DNA. The difference here, and it's not in this slide, is that you don't need helicase. Remember, helicase is the enzyme that unzipped DNA when we were just copying DNA. Well, there's a new enzyme that does this. That enzyme is called RNA polymerase. It's a fitting name because we're making RNA now. So it's not DNA polymerase anymore, it's RNA polymerase. And it also doubles as helicase. It's that special. It does both jobs. It does both jobs. So we no longer need helicase to unzip DNA if we're making a messenger RNA strand, right? RNA polymerase also assembles the nucleotides in the messenger RNA strand. So it's really the only enzyme you need to know for this process. Once that strand is made, the RNA separates from the original, oops, I don't know how that happened. Go to the current slide. RNA separates from the DNA, and it leaves through the nucleus. This big purple ball over here on the right is supposed to be the nucleus, and these holes in the nucleus are called nuclear pores. A pore is just a hole. So it exits through that pore into the cytoplasm and goes off to find a ribosome. So here's an image of it, right? In the nucleus up here on the top, here is your DNA. I'm going to go ahead and label this. I don't know why it makes this line. There's your double helix DNA. This yellow ball right here is RNA polymerase. It's unzipping DNA and building the mRNA strand simultaneously. It is that awesome. So that is RNA polymerase. I'm going to abbreviate. And this yellow strand here coming off is the messenger RNA molecule. And the environment, this big purple ball, is the nucleus. And obviously these little holes are the nuclear pores, right? So this is transcription. And then in this bottom picture, what they're showing you is the messenger RNA exiting the nucleus. And it's going off and it's finding a ribosome. That takes us to step 2, translation with an L. Here, you're going from the messenger RNA we just made to a protein. And it happens at a ribosome. So here are the steps. Ignore this should be a 4, that should be a 5. The numbers don't matter. It's just what happens is what's important. So messenger RNA travels from the nucleus to the ribosome. At the ribosome, messenger RNA meets up with tRNA. Remember that tRNA is carrying the amino acids so that they can be assembled at the ribosome. The anticodon and the codon match, which is how the tRNA meets up with the messenger RNA, drops off its amino acid, and then this just repeats. It repeats over and over and over and over and over and over again until you have what's called a stop codon. And the process stops, and the baby protein is released. So here's some images that should help. All right. So once transcription is done, here's the same picture we just saw. Here's the nucleus, here's the messenger RNA leaving the nucleus and going off to a ribosome. So in this bottom picture, and actually this ribosome should be one spot bigger, it's 3 codons wide. So this blue ball right here is the ribosome. Actually, the ribosome has 2 parts. There's this bigger blue part here that's colored in blue, and then down here, this kind of beige color is like the smaller part. You don't have to know that, but basically, there are cell signals that tell the ribosome to assemble. It's like ribosome, assemble, and the blue larger part here and this kind of beige smaller part come together to form the actual ribosome. And in real life, they're not that color, but they're that color just for this picture, right? So that's the ribosome. And again, that's made of rRNA. So here is the messenger RNA. This messenger RNA strand is the same one that's up here in the top. And at the ribosome, here comes your tRNA molecule, right? There's your tRNA molecule. On the top of it is an amino acid. It has a name. This particular one's called lysine. And at the bottom end of the tRNA molecule is the anti-codon, right? And look, it's going to match. It matches the codon that's on the messenger RNA, right? UUU matches AAA, right? They're complementary to each other. Same with the ones that are already in the ribosome, right? Here's an anticodon here that matches the codon AUG. This anticodon matches this codon, right? And the whole point of them matching is because they're specific, right? UAC and AUG is specific to methionine. AAG and UUC are specific to phenylalanine. That's how the cell, quote unquote, knows which amino acids it needs. It's all based on the codon and the anticodon matching up. So here's how the protein is actually assembled. There are 3 sections to the ribosome. There's the E section, there's the P section, and there's the A section. You don't have to know this. This is kind of a little bit of additional info, but you should understand the process, right? So the tRNAs enter at the A region, right? The anticodon and the codon match, and then the ribosome scoots to the right in this picture. It actually, the whole ribosome moves to the right. And then another tRNA comes in. The base pairs match on the anticodon and the codon, and the amino acid is dropped off. A third one comes in, and by this point, all 3 sections are full. So the one that's now on the left leaves, it exits. You can think of E for exit. E for exit. And it goes off and it finds a new amino acid to bind to, right? But basically what happens is they just keep on entering on the A side, and then the tRNAs leave on the E side, and the growing protein is made in this middle portion, this P portion. So what you end up with is a growing polypeptide, a growing protein, coming out of the P region, the middle region of your ribosome. And I will post a separate video that shows this all in motion. I know it's hard to visualize, but the video that actually moves and you can see it moving will help visualize this process, right? But in terms of quizzes or tests or things like that, you have to know the key players. You have to know what they do. And you should be able to identify any of these if I gave you an image. Right. So just a quick summary. Protein synthesis is made of 2 steps: transcription in the nucleus and translation at a ribosome out in the cytoplasm, right? The DNA molecule contains the information needed to build the protein, but because it's trapped in the nucleus, you need RNA to help. So your original DNA strand in the nucleus is going to be transcribed in transcription to a messenger RNA strand. Notice that all of the T's are replaced with U's. 3 bases at a time are now called a codon. When this messenger RNA strand moves out into the cytoplasm and finds a ribosome, it gets translated into a protein. Each codon codes for a different amino acid. They are linked together at the P site of the ribosome, and the baby protein or the polypeptide is formed. So the last thing is, how do we know, or how can we figure out which codon requests or is paired with which amino acid? So this is called an mRNA codon chart, and you have to be able to read this, and the good news is it's very easy. So remember, this is a messenger RNA codon chart, and I'm going to draw your attention, 17 stars here. A lot of students forget that this is an mRNA codon chart. This is not a tRNA codon chart. This is specifically for messenger RNA. This is for the codons, not the anticodons. So here's how you read it. You start in the middle and work your way out towards the edge. So let's look at AUG. If that's your codon, you're going to start here with A, then go to U, and then go to G. Hey, that codon pairs up with methionine, which is a type of amino acid. It's one of your 20 amino acids, right? This is what's called the start codon. In virtually not all proteins, but in most proteins, methionine is the first amino acid. Therefore, AUG is usually the first codon in a messenger RNA strand, and therefore it's called the start codon. Let's do some other examples. Let's do UCA. So here's U, work your way out. There's C, work your way out again. There's A. That's serine, right? And you can do the rest if you want. Some interesting things about this codon chart. What you're going to notice is there are redundancies. Certain codons get you the same amino acid. So let's look here. Let's go over here to G, C, G will get you alanine. So will GCA. That also gets you alanine. GCC gets you alanine, as does GCU. So alanine has 4 different codons that it corresponds to, right? It's not 4 for all of them, like aspartic acid right next to it only has 2 codons, GAC and GAU, but the point is that there are redundancies. So if, when we talk about mutations, there's a mistake in this last letter, maybe the original codon was GCC, but then it mutated into GCA. Well, fortunately, that change will have no effect because the overall product, the alanine, is the same. So some mutations, which are called silent mutations, don't affect anything. And we'll talk more about that next video. The last thing on here is if you look at this codon chart, you'll notice the word stop. Stop is not an amino acid. Stop is the signal that the ribosome knows that it's done. There's no more protein to make and it can release the protein, right? So there are a couple of stop codons. UAA and UAG are stop codons, as is UGA is also a stop codon. The only thing I'm going to ask that you remember is the start codon AUG, OG, and its associated amino acid, methionine. The rest you can always just reference the table. This codon chart is the exact same. It has the same information. It's just structured differently. So in this codon chart, you start here on the left where it says the first base, then you move, so you do the rows first, then you do the columns, and then you do this right side. So let's do A, U. Let's see what happens. So A, we're somewhere in this row. A, we're somewhere in this column. And when you go down and you find their intersection, they intersect right here. And then U, U is right there. So come over and ASN. Now this doesn't show you the full name of the amino acid. It just shows you the abbreviation. And like on a quiz or something, if you just put the abbreviation ASN, that would be perfectly fine. Let's just check and see where our start codon was. We know that start is AUG. So let's do it. Here's A, and here's U, so they intersect in this box. And then G is over here, so bring it over right there, MET, right? That's the abbreviation for methionine. So same chart, different format. You should understand how to read both. And here's a final overview. DNA to mRNA is transcription, it happens in the nucleus. Messenger RNA to protein is called translation, and it happens at the ribosome out in the cytoplasm. Right. So I hope this helped. I will post more videos on this for you to look at, and I will see you next time.